Now I'm replenishing energy reserves. Now I'm going to store fat for later, because I'm going to need it for the next time I see a tiger. Woo! This is a busy slide. This summarizes the whole thing. And your book would have a slide similar to this as well. A nice summary slide for your purposes. I can't, it's too small to walk you through. But we've made these observations in bits and pieces leading up to this kind of global slide about CNS, PNS, autonomic motor, sensory, and so on. All right, and that kind of, again, introduces us to this idea of the nervous system and its overarching architecture. How is it organized? From there, we want to zoom in. So we just looked at the forest, and now we start to look at some trees in the form of what is nervous tissue composed of? You should remember there's 4 types of tissues. Do you want to practice? What's the first one? Nervous, okay, I'll take that. What else? Muscle. My heart rate's going up.
Connective.
Connective and epithelial. Oh boy. That's basics, gotta remember those. You're gonna see that in the final, by the way. 4 tissue types. I want to remember the tissue type because I'm going to need to know that when I get to AP2. Epithelial, connective, muscle, nervous. I'm not upset if you don't remember. I'm just making an observation about I need to practice. Okay. So I can see some general things. This is a very cellular tissue type. Lots and lots of cells here. There are 2 main types of cells, of which there are multiple subtypes. Are there lots of different types of neurons? Yep. Are there lots of different neuroglial cells, or what are just called glial cells? Yes. Okay. And I can imagine, because I've written a question already like it, name and briefly say what each of the different cell types' function is. So as we walk through, you might think about making a little list for yourselves. Neurons, what do they do? Microglial cells, what do they do? And so on. Okay. So briefly, we'll start with the neuroglial cells. The first one's there at the bottom. It's called an astrocyte. Neuroglial cells generally are thought of as support cells for the neurons. Glial cells. So we don't always say neuroglia. That is very quickly shortened to glia. There's a journal called Glia. For the glial cell people, this is like the journal. I love it. Everything you've ever wanted to know and publish in the research world about glial cells. Again, they are thought to, they outnumber the neurons, you can see there, by about 10 to 1. And they are thought to mostly provide support for the neurons until recent. And there was a big fancy paper published in Nature, which is like one of the top journals. Science and Nature are the 2 best journals. We publish Science in America, Nature published in Great Britain. They found out that astrocytes did way more than some of these lower dots there. So during development, astrocytes provide the migratory pathway with which neurons walk across to get where they're supposed to go. They also function as nutrient processing and nutrient transfer. They're like the wet nurses for the neurons. All the busy work, they do it. They buffer sodium and potassium. Remember, too much of these ions is detrimental to that action potential thing. They buffer calcium. Calcium also deadly to neurons. And other neurotransmitters like glutamate also has been shown to be it drives toxicity in the brain. Now I'm thinking about MSG. You've heard of MSG? Monosodium glutamate, MSG. Was a major food additive for a long time. And now on all the labels it's advertised, no MSG. Why aren't everyone so fussy about MSG? It was found to be what? Neurotoxic. Whew. I'll wait till you take me for, I'm in my nutrition mode now. It's time to do nutrition in AP2 in my survey class. And this is when I get tizzied up about stuff, more than usual. Be careful. They put MSG in everything for a long time. Why? It tastes like meat. When we get to special senses, I'll talk more about this in a couple weeks. So it's on topic, but we're early. MSG is the taste of meat. The fancy word for the taste of meat in your mouth is called umami. Discovered in Japan, a big finding. You taste glutamate. When you eat meat, you're eating amino acids. One of them is glutamate. You have a sensor for this in your mouth. You need protein in your diet, so meat tastes good. We're programmed to like it. They found out that they can make a molecule. They can attach a sodium, one sodium to a glutamate, hence the verbiage monosodium glutamate. And you can put it in anything, and it makes it taste yummier, meatier. And then, oops, they found out it makes your brain die. And now, they don't put it in anything. But my point about the new finding for astrocytes was that they do information processing too. This was a big deal. I told you this a minute ago, right? If you want to go into a field where there's new discoveries being made all the time, this is one of the fields, neurology. Wow, that was a huge big deal. We went there circuitously, but this was the Nature paper I was trying to talk about at the beginning. When they found out that astrocytes send signals. This is a Papa Champagne kind of moment. We used to think that only neurons sent and received signals. Now we know astrocytes do it too. Interesting. Maybe if I have more astrocytes, I'm going to be way smarter. Right? I don't know. We'll see. They look like this in terms of their orientation to other cells. Astrocytes have processes. The verbiage here is they have these extensions. They're called processes. They have a process would be one, multiple process. PS at the end, processes. That extend out from the cell body. The cell body now is called a soma. S-O-M-A. Why do we make up all these new words? I don't know. The many of you thought you knew, changed. So these processes extend from the soma. They're part of the cell, right? And they do what? Wrap around neurons, and they wrap around blood vessels. You can see that there. Astrocytes form part of the blood-brain barrier. You've heard of this probably. BBB. Astrocytes form the brain side of the blood-brain barrier. They didn't call it blood-brain barrier for nothing. There's a double barrier. A blood side has a barrier, and the brain side has a barrier. The brain side of the blood-brain barrier is the astrocyte. Okay, next. Microglia, or what are called microglial cells. What's a microglial cell? It's a white blood cell. It's a modified version of a dendritic cell or a macrophage. It is a white blood cell. This is an immune system thing, right? The brain has its own resident population of white blood cells. They're called dendritic cells. Sorry. They're called microglial cells. They are a type of dendritic cell. Why is it important for fun? Why is it important that the brain has its own population of white blood cells? Because why? To fight off infections. But why can't they just use all the other white blood cells in the body? How are they going to get there? I just mentioned there's a blood-brain barrier. The job of the blood-brain barrier is to do what? Keep stuff out, right? I want to keep infectious stuff out. I also want to keep drugs, metabolites, heavy metals, lots of stuff out. I need a brain. I protect it, extra special than every other organ, with this fancy barrier. If I'm a drug discovery scientist, one of the first things I have to do, if I'm like a medicinal chemist or something, a pharmacologist, whatever, if I want to have a drug that's going to act on the brain, it has to do what? Cross the BBB, right? This is not easy. Most things don't. So white blood cells don't cross either. Not easily, not under normal circumstances. And there's other stuff that has to be removed regularly. White blood cells not only fight infection, like you were aware, they just clean up messes. In general, white blood cells clean up messes. Yes. They know the structure now of, we know how to modify molecules to make them cross the BBB now. So there's been enough of this medicinal chemistry done that we've figured out what the chemistry is, the structural chemistry of a molecule that crossed the BBB. And we can just attach these little moieties, these shapes, onto pretty much anything, and they'll get in. And MSG crosses easily because it's a small molecule. There's other limitations to the drug discovery process that are liver-related, but not relevant to this class. Okay. And the third cell there, ependymal. So I'm making a list, right? You're making a list. So far, so good. This is the easy part. Ependymal cell is the next one. You can see there what the ependymal cell does. And there's histologies. You already looked at the slide on the right for both of these, how they look. Ependymal cells. They line the fluid-filled cavities in your brain and spinal cord. Your brain and spinal cord has fluid, your brain and spinal cord is suspended in fluid. What's that fluid called? CSF, right? Cerebrospinal fluid. You've heard of this. You've heard of this. And the fluid does what fluids do. Provide protection, provide support. They also provide buoyancy. This is interesting. The cellularity of your brain and spinal cord is such that it would crush itself under its own weight if it wasn't suspended in fluid. Interesting. What that means, I don't know. It's just true. There has to be fluid there. Your brain and spinal cord literally float inside of this fluid. Without this fluid, it wouldn't work. These ependymal cells help to make the fluid, and they help to circulate the fluid. They are ciliated. This is a type of epithelial cell, by the way. Does it say that? Yeah. It's a columnar epithelial-like cell. It's ciliated. It sweeps, and it drives CSF flow through the fluid-filled cavities, which are either called canals or ventricles. Depending on what, so ventricles are in the brain, and the canals in the spinal. We'll add those words in. The simple, what I need you to be able to put for the test is they circulate the CSF, and they help to make the CSF. Okay. Oh, don't worry about this. I like immune system stuff too, so I put some things that's too much. No, don't worry. Okay, there's the cartoon for ependymal cells. Okay, last two. Number 4, oligodendrocyte. Oligodendrocyte. This is often just called an oligo. Short. No one wants to say oligodendrocyte all the time, right? When people research these are extremely interesting cells. And the reason why is written on the slide. Oligodendrocytes produce a lipid wrapper that goes around the axons of some neurons. Oligodendrocytes produce a lipid membrane wrapper. Look at the cartoon. The oligodendrocyte extends a process. It's part of the cell, and it just wraps itself around and around and around and around, over and over and over again, on part of a neuron called an axon. Do you see that from the cartoon? The stuff that they made, this wrapper, has a name. It's called myelin. M-Y-E-L-I-N. It's called myelin. It's a bold word there. The whole thing is referred to as the myelin sheath. The entire length of it is called a myelin sheath. It is composed of multiple segments of myelin. So there'd be myelin segment, would be one segment. Myelin sheath would be the whole thing. I'm going to finish with, not yet, I'm going to finish with this concept, okay? This is important. And you're going to see in a minute, there's another cell that makes this stuff too, and it's different, so I want you to pay attention. The other stuff so far didn't have to concentrate on it. Now I need you to concentrate. In the CNS, in the central nervous system, make a note, the cell is called the oligodendrocyte. So the oligodendrocyte is only in the central nervous system, only. It makes myelin. Myelin is an extension of the plasma membrane of the cell. Plasma membranes are made out of lipids. Fats are insulators. This is an insulation. The myelin sheath, simply put, is an insulator on the axon of the neuron. The axon of the neuron is like the electrical wires. So how can we translate this to something you know about? This will all become clear, I promise. So this translates very easily to the way the building in your house or whatever is done. Do I want to have all the wires where they would crisscross and lay on top of each other in a big pile? Probably no, right? Wrapped around each wire is a cord, right? That insulates and protects the wire. Piping and heating and cooling pipes are the same way, right? They're insulated. If the wires are crisscrossing, then the signals are going to get crisscrossed, and they could be sparks, and we could have a fire. The same thing would happen in your brain. I need the wires on the neurons, it's called an axon, to be separate from all the other wires. How do I get them all to be separate? I put a little insulating wrapper on them. What's it called? Myelin. What cell makes it? Oligodendrocyte. In the CNS. Now, look at the cartoon. How many myelin segments does one oligodendrocyte make? Lots. Don't count them up, it's a cartoon, right? Lots, okay? More than one. Okay? You're good with CNS myelin, right? Myelin is an insulating wrapper around the axon. And one oligodendrocyte found in the central nervous system only can make a lot of different myelin segments. That's different than number 2 on the list for the PNS. So the next cell is called the Schwann cell. If you're making a list, the next one is Schwann cell. It's capital S because it's named after the person who described it first. We'll go back to that. Satellite cells are boring. We don't even know what they do. Next. Seriously. I told you there's lots here we don't know. The other interesting cell for the story is called a Schwann cell. What does it do? It makes myelin too. Where? PNS. So I've got 2 cell types that do the same thing. One in the CNS, one in the PNS. So in the PNS, it's called a Schwann cell. It makes myelin, just like before, which is an insulating wrapper around axons. But notice now, how many segments of myelin does each Schwann cell make? just one. In this case, and here's where it gets a little confusing. In this case, the Schwann cell itself is the myelin segment. The Schwann cell is the myelin segment. One Schwann cell equals one myelin segment. The cell itself, instead of sending out processes that touch lots of cells, it just wraps itself around, and that's all it does. So there's way more Schwann cells than there are oligodendrocytes. Now, you might be sitting there saying, why is it like this? Why do we have separate cells that do the same thing? And this seems less efficient. It'd be easier to have one cell that can make lots of different segments than just one cell, one segment, right? That's a question for a different day. We don't know why. But we do know, and here's the interesting part of the story, which is where we're wrapping up today. The Schwann cell myelin is different than the oligodendrocyte myelin. Again, the metaphysics of why we don't know. We never know. But we do know some important things. And one is that the myelin produced by the Schwann cell is significantly different than the myelin produced by the Schwann cell. That is fascinating. And if you're not convinced, I'm going to tell you why it's fascinating now.
Because it's really good.
Does the central nervous system regenerate on its own? If you fall off a horse and have a spinal cord injury, are you going to walk again?
No. Well,
Probably not. You're in a major car accident or whatever it is, right? Break your back. You're probably not going to walk again. Now, of course, you know that with modern technology and science, welcome to science, and researchers like this, now your chances of walking again are really good, much better than they were. Again, before it was like, oh, well, here's your wheelchair. Not anymore. Not anymore. Does the PNS regenerate naturally?
Yeah.
Yes. Listen, this is important, and I know it's on the test. Does the peripheral nervous system naturally have regenerative capacity? Yes, it does. PNS regeneration is facilitated by the Schwann cell. The Schwann cell drive it. They regulate it, they control it, they help it along, they promote it, whatever words you want to put. PNS regeneration is driven by Schwann cells. They secrete factors that promote the regrowth of axons and the regeneration of nerves. It turns out also that when there's an injury, there's damage, right? The debris left over from a Schwann cell peripheral nervous system, like a crush injury or something. The debris left over promotes regeneration. The myelin itself, the debris of the myelin, the leftover garbage, promotes PNS regeneration. Interestingly, it's exactly the opposite in the CNS. Why doesn't the CNS regenerate? Because after a crush injury, or whatever you want to call it, the damaged oligodendrocyte myelin is inhibitory to regeneration, literally blocks it. We know this because we can take the proteins and the factors, we know what's in myelin, and we can put it, I did this in the lab, we can put it on a little slide, and I can put neurons here, and I can put some neurons there, and I can put Schwann cell myelin over here, and oligodendrocyte myelin over there, and watch this side regenerate, and this side not. The axons will grow right up to the myelin sheath where I put a little sprig of it down, and turn, and go the other way. The axons want to regrow, that myelin blocks it. Kind of sucks, huh? Again, the metaphysics of this, we don't know. Why doesn't the CNS regenerate? Who knows? You could speculate about permanent wiring and all this crap. We don't know. But knowing how it works, what am I doing now to treat patients? Researchers and clinicians partner up all the time, right? Insights made by research scientists. Now, clinicians, we can make your spinal cord regenerate. We know the factors. We can inject them, right? We can clean up the mess. We can clean up the mess on a CNS crush. That helps too. Now we have nano scaffolds, and we can put a little mesh in there, full of what cells? Schwann cells. Hey, they promote regeneration. Why not put them in the central nervous system? Does the same thing. Should work. Guess what? It does. It does work. And now they have dogs that learn. You can watch a video, YouTube it. Dogs get hit by cars in big cities, right? There's a few centers in the US that does this work. Dog gets hit by car, take it to the clinic, clean up the mess, insert, Dog can't walk, 6 months later, dog walk. Wow. Why? Because we figured this out. Tracy?
Can I ask if, there was somebody who recently had a stroke, and they gave a stem cell transplant into the brain?
Yep. That's another thing we can do now too. Put some cells. Okay, that's what I have for today. Again, some of that will be on the test, right? But mostly I'm trying to pique your interest, right? This is cool. Okay, have a nice Tuesday. Don't forget to study. Test next Thursday.
No video?
We'll watch the video on Thursday.
Okay.